Okay, I think we slowly get started, although some people seem still to join in. So welcome everybody to this session, where we have a number of talks in the area of nucleic acids for delivery or nucleic acid delivery and for targeting, and essentially as therapeutics themselves. And we believe that this was a good choice to introduce here because nucleic acids might be among the future emerging nanodrugs. So again, welcome everybody to this session, and I would like to introduce the first speaker, who is Professor Paul Anderson from MIT Harvard, or Harvard MIT, and he will report about his approaches in siRNA delivery nanoparticles. So thank you very much for coming. We look very much forward to your presentation.
Thank you. First, thanks very much for the... Oh, sorry. Thanks very much for the invitation, and really appreciate coming here. So I thought what I would talk about today is our work on the development of RNA nanoparticles. And just generally as background, it's increasingly clear that the intracellular delivery of biomolecules has tremendous therapeutic potential. So there are actually quite a growing list of molecules that if we can just get them inside of cells, they really can be therapeutic. So whether that's RNA in the form of siRNA or messenger RNA or microRNAs, DNA in various forms, and even some proteins. And so what I'd like to start talking about first is our work in the area of siRNA, and just to provide some background. So in 2006, the Nobel Prize was awarded for the discovery and characterization of this pathway, the Rna Interference Pathway, which turns out to be really a fundamentally important process by which all of our cells are regulating gene expression. And so at a high level, what happens is double-stranded RNA is broken up into small pieces of around 20 nucleotides. And then these double-stranded RNAs can then insert into a process called the RISC pathway. And once loaded onto this complex, can cause the catalytic degradation of the complementary messenger RNA. So this is a process by which messenger RNA can be catalytically destroyed depending on the sequence of the RNA that's loaded. And so it wasn't long after the discovery of this pathway that people started realizing that there was tremendous therapeutic potential. The idea is if you can get a double-stranded RNA inside of a cell, you can potentially interact with this pathway and specifically turn off any gene of interest. And the reason that's exciting, if we just sort of think about RNA interference relative to conventional therapeutics. So conventional, so I think there's this term that we're starting to enjoy using called modular pharmacology. And so the idea is basically that with essentially the same drug, just by tinkering with the sequence of an RNA, you can turn off any gene in the cell. And so I think there's tremendous excitement about being able to basically have one drug where we just alter it slightly and now have a completely different therapeutic effect. And so the fundamental challenge really is, how do we get RNA to work as a drug? There's no question that the biology behind this process is solid. And so when we think about RNA as a drug, it actually has a lot of challenges. So first of all, RNA is a large molecule. It's highly charged. It doesn't cross cellular membranes. RNA is prone to nuclease degradation, and there are a variety of processes in the body that can lead to immune stimulation as a function of RNA binding. And so that's really the first level of challenges around RNA, but also when we think about delivery, it's actually a multi-step process whereby we envision a nanoparticle, for example, entering the bloodstream, traveling to the target tissue, extravasating to our cells of interest, and then entering the cells typically through a process of endocytosis. So once in a cell, it needs to escape the endosome. If it's in a nanoparticle, it needs to decomplex. And then ultimately, if it is a DNA, it needs to get to the nucleus. And so this, really the delivery challenge is very significant here. And when we think about the process of turning RNA into drugs, there are really sort of three key pieces, and I'm only going to talk about one of them. So the first is sequence selection, and actually there's a whole lot that's known about how to pick the right short interfering RNA to get the best potency and specificity on your target. Secondly, there's really a very exciting amount of work on chemical modification of RNA, both to prevent nuclease degradation, to increase potency, to block immune stimulation, and now actually there's some very exciting human data showing that the connection of certain ligands directly to RNA can lead to therapeutic effect in the liver. But what I'm going to spend my time today is really talking about nano encapsulation, since that is what the meeting's about. And so nano formulations really are currently the most clinically advanced form of RNA drugs. And it looks like they're going to be particularly important when we think about long RNAs, where some of these chemical tools just aren't available right now. So when we first thought about finding the right materials to deliver RNA, we were naturally inspired by all of the great work that had been done in cationic lipids. And so here are just a few of the structures of the lipids that people had worked on for many years. When we first started working, we felt that while cationic lipids had tremendous potential, perhaps the chemical diversity of the space could be broadened. So for example, if you look at DOTMA, this is a lipid from the 1980s. DLIN-DMA was published in Nature, I believe, around 2006. And they're really only different in a few carbons, and that's not to take away from the work by Protiva in developing this material, but rather just to say that perhaps there are other chemical structures that could be useful for delivery that hadn't really been explored. And so the first question really is kind of a chemistry question. How do we increase the chemical diversity of cationic lipids? And so if we think about the synthesis of a conventional cationic lipid, they're typically multi-step processes, and this actually isn't all that hard from a chemical perspective, but if you wanted to do this 1,000 times or 2,000 times, and each time you did it you had to customize it, actually becomes very challenging. And so I only want to show a small piece of chemistry, but this was really the beginning of our efforts to try to find rapid ways to make large libraries of lipid-like materials. And so this is a Michael-type addition between a acrylate and a amine, and when you do this in one step you end up with this structure. So if you can compare this to a conventional cationic lipid, the orientation of the esters is different, the linkage point on the amine is different, but we felt that this might be worth further study. And so since this early work, we've now gone on to make thousands of these, what we call lipid-like or lipidoid materials. And with these materials, you can form nanoparticles with RNA and various other compounds. The process is basically taking a lipid-like material, this is one early material shown here, polyethylene glycol lipids, RNA, and then under controlled mixing conditions, you can end up with particles in a range of size, but typically we do a lot of work around 100 nanometers. So if you have thousands of materials and you can make thousands of nanoparticles, the first question really is how do you find those that are going to work? And one important workhorse assay for us has been this, which is an assay to look for RNA delivery to the hepatocytes in the liver. And the general idea is that factor 7 is a blood clotting factor produced in the liver. And you can create a nanoparticle with your material and factor 7 siRNA, and do a tail vein injection, and then just take a simple bleed, just take a drop of blood, and measure whether or not you got knockdown. And so this assay really allowed us to test thousands of materials, and we've gone through a lot of mice this way. But so at a high level, the process has been doing our chemistry to make libraries of materials. We try to automate these processes whenever we can. We take our lipid-like materials and we use controlled mixing, usually microfluidic systems, to make nanoparticles with other colipids. And then we inject these into animals and see which of those are giving us delivery. And so in the interest of getting to newer material and covering a range of topics, I want to just give a couple data points. So the first data that I thought I would share is a primate study that we did, where we took one of our lead materials and an siRNA or a control siRNA. We made a nano formulation, injected into non-human primates, and then when we look, after a single injection at really very low doses of RNA, we can see almost complete knockdown in the primates. So these are actually two different studies. And even at the lowest dose of around 0.03 mg per kilogram, we're getting very good silencing in the liver. And so this overall process has led to the discovery of a range of different materials. These are just two of the structures shown here. I didn't want to get into chemistry too much, but I'm happy to answer questions later. And we're starting to see human data with materials and lipid nanoparticles. And so this graph shows knockdown in primates relative to knockdown in humans with a similar material, MC3. And so what you can see is that in humans, you're getting good knockdown in the liver that after the first injection will last out to about a month. And that parallels nicely what's observed in primates. And so these studies not only get us excited to think about the possibility of using RNA in people, but also that these primate studies we do can be predictive of performance. And so over the last, say, 10 years, well, maybe no less, maybe 8 actually, there's been a tremendous improvement in the potency of nanoparticles. And this is just to kind of summarize performance of key lipid materials over the last, say, 8 years. So when we first started work in this field, basically took around, so sorry, what you're looking at is the expression of a marker, factor 7. So this is 50% knockdown when you hit this purple line, and then you're looking at the dose of RNA. So in the early formulations, it took over 10 milligrams per kilogram to see 50% silencing. And over the years, this potency has improved and improved over 3 orders of magnitude, where now we're at about 0.002 mg per kilogram in our latest formulation. This was published actually this year in PNAS by our lab, and we've participated along the way. Actually, these bottom 3 formulations are all in clinical development in various locations. So the potency of these formulations is exciting because it makes us think about the utility in humans, but it also allows us to start thinking about more complex diseases. So here's an experiment where we take 10 different siRNAs to 10 different targets, and we make a nanoparticle and do an injection, and after a single injection of this formulation, you can see silencing in all 10 of your targets. They're not all equivalent, but you see significant silencing in all 10 of these. So I think there's a perception by people that are outside the field that perhaps siRNA will only work in the liver, and I think, we think that's just not true. I want to give one example of where we think siRNA can also be useful, which is in the endothelium. So there are a variety of endothelial beds that we think could be useful for therapeutics, but the real question again is how do we get RNA into endothelial cells? And so I'm going to talk about some new materials that we've developed. They're short amino polymers, and the overall process has been first similar to the lipid procedure. We do our chemistry, we make libraries of materials, we do controlled mixing with RNA, and then instead of looking in the liver, we're looking at RNA delivery to the endothelium. And so we've identified some materials that with RNA can form very small nanoparticles of around 35 to 55 nanometers. Actually, they're EM. They have this nice multilamellar structure. It looks sort of like gobstoppers. It's a very reproducible process, and the particles are stable in storage for many, many days. So with this formulation, if you have an RNA targeting a gene in the endothelium, and you inject that IV into mice, you can see silencing in vivo in a range of different tissues, of endothelial tissues. So again, what you're looking at is knockdown of ICAM2, which is an endothelial gene, and you're looking at a decreasing dose of RNA. So you can see at the highest doses, we're seeing silencing in the lung, in the heart endothelium, in kidney, and also in liver. And as the doses become lower, we see reduced silencing, but in particular in the lung, we still see significant silencing. And so right now, this is a very potent formulation for lung endothelial delivery. And so we can take 5 different siRNAs to 5 different targets, make a nanoparticle, inject this into mice, and then look in the lung endothelium. And so what you're looking at here is the 5 different targets with decreasing RNA. And so even at the lowest dose of total siRNA, we're seeing silencing in all of these targets, showing the potency of the system. And what's interesting about this process is not just that we're able to get silencing at the endothelium, but in fact the tropism of these particles has changed. And so when we look at our liver formulation versus the endothelial formulation, what you can see is the endothelial formulation, as I just mentioned, you're looking at the amount of RNA needed to get 50% knockdown. So here we have 0.02 mg per kilogram. With our liver formulation, we really don't see any silencing in the lung endothelium. In contrast, in hepatocytes, the liver formulation is very potent, 0.01 mg per kilogram to get silencing, and it takes a very high dose of the endothelial formulation to give us any silencing in hepatocytes. And so just by changing the chemistry, it's actually possible to change where these nanoparticles are silencing. So it turns out, so I'm in the Koch Center for Cancer Research, and our director is known in particular for his work in lung cancer. And so we've been investigating whether or not these systems that can deliver RNA to lung endothelium could have utility in lung cancer. And so we've been working in a genetic model of lung cancer called the KP model. It's based on a KRAS mutation, P53. And basically, this genetic model causes the induction of tumors, small cell carcinoma tumors in the lung, and recapitulates, at least to the best of the mouse model ability, what is seen in patients. And so, knowing the biology of these systems, we realized that we wanted to first deliver an RNA to silence KRAS, and then also we wanted to promote P53 signaling indirectly by delivery of a microRNA. So I'm not going to go into the biology, but basically by studying the genetics of this cancer model, one could rationally design targets that could perhaps interfere with it. And so the question is, first, can we make a nanoparticle of these RNAs? Can we get RNA into the tumor cells? Can we see slow in tumor growth, and then and also possibly a combination with conventional therapeutics? So first of all, we can deliver to these tumors. So this is just showing knockdown of luciferase over time following a dose of a formulation targeting luciferase versus the control. If we deliver microRNA 34 formulations or KRAS formulations, we see a very significant reduction in the growth of these tumors. And in fact, when you combine these, you actually start to see regression of the tumors. It's a very tough model. So what you're looking at is the growth of the tumors over time following a dose of the nanoparticle with miR34A and KRAS versus the control RNAs. And so these formulations can be used synergistically with conventional therapeutics like cisplat. And so when we look at experiments where we do a series of injections and then stop, comparing cisplatin to the RNA therapy to a combination of RNA and cisplatin, we see a very long extension of lifespan past the last injection in this difficult model. So we're optimistic that these types of formulations may have potential utility in lung cancer. There are also certain immune cells that can be reached with siRNA nanoformulations. And although there are many immune cells we're interested in right now, it's only certain leukocytes that are accessible to these lipid nanoparticles. And so the experiment, again, I want to show you is a RNA targeting CD45 in a nanoparticle given to primates, and then looking in the peripheral blood to see if we can get silencing of certain leukocytes. And what you can see is actually about 65% knockdown of all of the peripheral blood leukocytes in this primate following a single injection. So right now, the nanoparticle delivery options available to us are sort of captured here. We can now hit all of the cells in the liver, and this has been shown in rodents and primates, and now also humans for hepatocytes. The endothelium of many organs we can access, certain leukocyte populations, and there's a growing list of tumors that, at least in rodent models, have shown utility. You know, siRNA isn't the only form of RNA of interest. As I mentioned earlier, another sort of half of modular pharmacology is turning genes on. And so the idea here is, instead of trying to turn a gene off with RNA, if we actually deliver messenger RNA, we can turn genes on and produce new proteins. And so when we think about the comparison of delivery of sRNA to mRNA, it's actually even more challenging with messenger RNA. So while sRNA is a large and double-stranded, which provides some small protection from nucleases, mRNA is largely single-stranded. sRNA, there are a huge number of chemical tools available to do total synthesis at a pharmaceutical scale of short interfering RNAs. Messenger RNAs that need to be much longer, actually there's a limited set of chemical tools available, so it's harder to create nuclease-resistant and delivery-promoting structures chemically. And while sRNAs are big, mRNAs are really, really big. I mean, up to 1 million Daltons or more, so much larger. And I think in general, at least in the near term, the only way to deliver these systemically is going to be through Nano Encapsulation. But one can take the same types of materials that we used for sRNA and used them in slightly different ways with messenger RNA. So again, taking a lipid material, colipids, messenger RNA, controlled mixing, making nanoparticles. We actually got started in this first in vitro, and we were interested in using messenger RNA for reprogramming of cells into stem cells. And so we actually find with messenger RNA in vitro, we can get huge transfection to a range of different cell types. This is using a GFP message, and in fact, this material has been commercialized for this process. But what we're really interested in is, can these materials be used systemically? So the first mRNA I want to talk about is for erythropoietin, so EPO. You may know, if you're a cyclist fan, from Lance Armstrong, but also it is an important drug for anemia. Basically facilitates the production of red blood cells or the bone marrow. So the experiment here is we take a modified RNA encoding EPO. We make a formulation, inject it into rodents, and then we're looking for soluble human EPO protein in the rodents. So after a single injection of a nanoformulation with these materials and messenger RNA, we can see a huge spike in human EPO, actually about 60,000-fold the physiological levels that you would normally see in a person. And this is reflected in a dramatic increase in hematocrit. Their blood becomes viscous. And in fact, recently we've shown that the same delivery potential is also possible in primates. And again, because this is modular, we're not limited to just using one target. We can, by changing the sequence, create a drug for a different target. So here's an example where we take a message encoding factor 9. And I'm not going to step through the factor 9 pathway, but this is an important protein for blood clotting and hemophilia. So we take our message for factor 9, make a formulation, inject it into mice. And this isn't looking for EPO, we're looking at factor 9. And again, about 24 hours post-administration, we can see very high levels, about 100-fold past sort of where you would be considered severely deficient of human factor 9 in these mice. So in the last few minutes, I wanted to talk about even more complex delivery challenge. So we're excited by the opportunities to deliver RNA to turn genes on and turn genes off, but what about fixing genes? And so there's a growing interest in the idea that perhaps some of these specific nucleases can be engineered to allow correction. The newest of these is called CRISPR, and this is basically a system whereby you can, if you get it into the cell, it causes specific cleavage at whatever site you want. That can be engineered, and then through homologous recombination you get repair. And then so now the idea is not just causing a transient effect, but perhaps causing a permanent change to the genome of an animal. And there are not just CRISPR, but also things like TALEN and zinc finger. So there's a range, a growing list of proteins that can cause these specific nuclease sites and be used for targeted repair. So I don't want to actually step too much through this, but the overall idea here is that there's a nuclease, and then through homologous recombination, you can get precise gene editing. And so again, when we think about this from a delivery perspective, we can compare it to siRNA. We know our siRNA is a large double-stranded RNA with lots of chemical tools. For this system, we need actually a little bit of everything. So we need to get in the nucleus of our target cell. We need a protein called Cas9. We need a guide RNA that that allows for specific cleavage. and we need a DNA that is the repair template that allows for correction. So we need a little bit of everything inside the nucleus of a cell, even more complex delivery problem. And so we've been looking at a model system it's a genetic disease of tyrosinemia that leads to liver and kidney damage, and it's actually about 1 in 100,000 worldwide and greater in Quebec. And it's a simple point mutation in people that causes this profound genetic disorder. And so in our first study, which we published earlier this year, we showed that a high pressure injection can actually lead to repair, to sort of delivery of the CRISPR complex, all three of those components, into the hepatocytes of a living adult animal. And so we were actually able to basically correct this genetic disease with a single high pressure injection. Now the challenge is obviously high pressure injection is not a preferred mode of therapy for people. And so we've been working to try to develop formulations that can allow us to replace high pressure injection, maybe more clinically suitable. But this is a complicated problem. So there's interest in using viral systems, but Cas9 is so large and really doesn't fit into the current clinically suitable viral systems, like adeno-associated virus. And so one clinical potential formulation might be using messenger RNA nanoparticles, delivering the guide RNA by a nanoparticle or a virus, delivering the DNA by a virus or a nanoparticle. And so we've recently been able to develop a formulation that gives us a correction rate of greater than 1%, which, while still low, actually is high enough to think about using clinically for some genetic diseases. So I think in the interest of time, I need to end there. I just want to thank a lot of smart people, Bethad Al-Nilam, a company I work with, MIT, in particular, many smart people, but also Shire as well. Thank you.
Okay, thank you so much for this very exciting presentation. I think especially from my perspective, the ability to have different siRNAs targeting different genes within one therapeutic particle is very attractive, you know, also in terms of drug resistance and tumors. Other questions from the audience?
Yeah, thank you for the really interesting talk. I have a question regarding the toxicity of these lipids. What is known about the toxicity, and do you have to do additional therapy with, I don't know, dexamethasone or something additionally?
Yeah. So the earliest formulations are associated, so first I'd like to say it's a function of dose. And so, you know, at the lowest doses, you really don't see anything. So these are doses where you see knockdown, but you don't see toxicity at all. As you increase dose, higher people have observed immunostimulation catalyzed by the lipids. And so that's why the early clinical trials did combine steroid therapy with the initial dosing. So newer formulations seem to be able to get around that. Another issue with toxicity with some of these formulations is clearance. So depending on the material, if you're envisioning treating someone every 2 weeks for the rest of their life, you worry a little bit about accumulation. And so there's actually been exciting developments developing biodegradable lipids where clearance is actually on the order of about 8 hours. So while certainly the earliest formulations have had challenges, I think there's been important developments recently.
Thanks.
Further questions? Please, in the back.
I think, is there any limitation in size of RNA molecule you can deliver using these lipid molecules, lipid system? I mean long non-coding RNA, of course.
I'm sorry, could you speak a little louder? I couldn't hear you.
Is there any limitation in size of RNA molecule you can deliver using this, by means this lipid system? I mean long non-coding RNA, for example.
Yeah, it's a good question. So I think it's too early for me to give a clear answer to that. We have delivered things up to 1,000 nucleotides. There may be some data suggesting that smaller RNAs are easier to get inside, as you might expect, but we haven't yet run into an upper limit where we just can't do it.
There was one more question in the back here, in the last row.
I want to ask, what is the advantage of using messenger RNA to deliver messenger RNA over the use, for example, of the conventional plasmid DNA to express the certain proteins within the cells?
Sure. Yeah, the first and actually most important advantage is that the current plasmid delivery systems are not very good for hepatocytes, the non-viral plasmid DNA systems. So I think that is the sort of first and foremost challenge. Secondly, there is concern that DNA as a drug could lead to recombination events and mutagenesis, and there's less concern that RNA could do that. I think we could debate whether or not that fear is warranted, but I think it is clear that certain regulatory bodies do have concern about the use of DNA as a drug.
I might have one short question. That is, did you evaluate how many siRNA molecules you have per liposome?
You know, we did, and I forgot the number. It's a couple hundred, I think.
A couple of hundred?
Yeah.
Okay. Okay, I think we would like to thank you again for this nice presentation.
Thank you.